Hello, everyone. We will be starting with the session in 10 more minutes. Thank you for joining the session. In the meantime, you can introduce yourself in the chat box and let us know about your educational background and your research background as well. And we are looking forward to it. Thank you. Hello, everyone. We will be starting. Hello, everyone. A very warm good morning, good afternoon, or good evening, in whichever part of the world you are. I would like to welcome you all to the very first session of Cheminformatics for Biomedical Drug Discovery program. This program is designed to address the challenges which are associated with understanding, modeling, screening, and applying cheminformatics strategies to improve drug discovery results. The process of finding a new drug against a chosen target for a particular disease usually involves high throughput screening, wherein large libraries of chemicals are tested for their ability to modify the target. With contributions from leading researchers in academia and biotechnology, pharmaceutical industry, as well as experts from the software industry, this comprehensive mentor-guided training program explains how cheminformatics enhances drug discovery. Today, we'll discuss about some of the topics which are mentioned here, which includes the molecular structure of biological macromolecules, history and techniques, amino acid structure and function, protein structures, primary, secondary, tertiary, quaternary structure, structural domain, structural motif, protein subunit. We'll also talk about protein-protein interactions, conformational analysis, protein folding, sequence to structure and function relationship, and we'll have a hands-on session with biomolecular visualization, and finally an assignment for you all to work on. So before we begin with today's session, as this is our first session, I would like to introduce us for those who have joined us for the very first time. We are a US-based bioinformatics company, and we are working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The OmixLogic training has been completed by over 24,000 participants from 187 countries in over 300 workshops in 6 different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and cheminformatics for biomedical drug discovery. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. Over the past several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The OmixLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Now, getting back to the program, let's discuss how do OmixLogic resources help in advancing bioinformatics. So the OmixLogic is a portal for practical and theoretical learning of bioinformatics, which includes various omics fields that we are covering, data science and structural biology. I'll be taking you through the portal and the resources and what all you can leverage from in just a while. But before that, I would like to go through some of the resources that you will have the access to as a part of this program. So the OmixLogic portal is composed of courses that are designed to take you through the theoretical knowledge. The OmixLogic takes a combination of training modules that I just explained, data analysis tools, curated project data sets, and interactive sessions with mentors will be conducted to give you a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, OmixLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic languages, basic coding languages like R and Python would also be given, and beginning that would help you begin a data analysis project. We will also combine pre-recorded mentor sessions, online live Q&A sessions, and self-guided study material, including courses on the OmixLogic Learn portal and example projects. which will include some practical assignments and quizzes as well for an immersive experience that has proven to be effective in our well-known and respected OmixLogic programs. So the program will leverage our top-rated online tutorials that I'll be just taking you through using the portal with smart tracking of activity and curated datasets designed for a great learning experience. Communication plays a key role for your activities in the program. So please make sure that all the participants of the program have joined the Skype group where you can access the session recordings, learn about the associated resources to be completed as a part of the session, get updated on supplementary material and assignments. You can also clarify your questions or request a live Q&A meet with the mentor for the same. And not only this, but you will also get to interact with our bioinformatics community and grow your network. And we have a note for the participants who have taken up the advanced subscription level. As a part of the advanced subscription level, you are supposed to work on a research project as soon as the program training is over. So you are requested to fill out the research proposal form as soon as you have your research question designed for the same. Now, what is there in the research proposal form? That is what I wanted to highlight on. So first of all, you have to mention your basic details, and then you have to mention the project title that you would like to work on, that you will definitely come up with after reviewing literature for the same. Then you have to provide a project description and then a project summary that should include the background of the literature review that you went through, what your research question is specifically, what are the objectives and how you want to answer the research question, like a flow that you have analyzed after reviewing the literature. And then you have to provide the link for raw data. Now, what is the link of the raw data here? It is definitely the protein structure that you want to work with. So you have to give the link to that particular protein structure or molecule, and then you give the references. We will share this project proposal form with you over an email as well. I just wanted to go through this proposal form with you once so that you know what you're expected to submit as soon as your training is complete. All right, so that were a few points that I wanted to discuss before we get started with the session. Now let's quickly sign up on the portal and make sure that we have everything ready before we start with the session and we have all things done for the program. I'll quickly share my Google screen now. All right, so those who have already created an account for themselves on the OmixLogic Learn portal, please put a yes or one in the chat box so that we know you are all set. And those who have not created an account yet, please do so. We'll be giving you a few minutes here so that you can create an account. Our team is here, and if you face any technical issues, we'll be able to assist you. We'll pause here for a minute or two so that you can easily create your account, and the ones who have created their account, please take this time to update your profile. I'll give a quick demo about how you can sign up on the OmixLogic Learn portal if you are new to this platform. So the link is in the chat box. Just click on the link. It will redirect you to the OmixLogic portal. You simply click on the login button, which will give you some options to sign in. Either you create some credentials for yourself and put in your details, which includes your name, your college or institute, and create an account that way. Or you simply use any one of your social media accounts, that could be Google, Facebook, GitHub, and sign up. That is create an account on the portal. I'll use one of my Google accounts to do so. Please feel free to ask us any question related to sign up for now if you are trying to sign up and not able to do so. Be awaiting the responses in the chat so that I know that we are all set to proceed ahead. Thank you, Miti Mathur. Great comment. What about others? Have you set up your profile? All right, let me just also give a quick demo for those who have created that account yet. So what's the next step for them? The next step for them is to update their profile. So please make sure that you have an updated profile picture. Your name should be in title case and add a brief bio about yourself that could include your educational background or your research interests. An updated profile helps the mentor know about your background, first of all, and also we'll be monitoring your progress and the backend for the program on your profile section itself. So this is the place where we get to know what you have been completing, which resources are you working on, which assignments have you submitted, which quizzes have you completed. And also a complete profile helps the recruiters see your profiles and maybe give you good, nice offers. So this is what a complete profile could offer you, where you can see your activity, you can see your completed courses, and also once a course is completed, you can easily download your certificate and share your achievements with the world. This is one thing that I wanted to share, that was about sign up and making sure that you have a complete profile. The next thing is that each and every step that you complete in your coursework or in assignment that you complete or quiz that you complete, your achievements will be recorded on our achievement board and we'll be able to monitor your progress from here. And achieving the specific points that are required for you to complete a good number of courses and your program could give a chance to you to appear on our leaderboard. So here is the leaderboard link. All these links could be found in the important links section that is towards the bottom of the portal. So here you can get a chance to appear on a leaderboard as well with an exceptionally great performance on the OmixLogic Learn portal. And also you stand a chance here to view the job listings that we are updating on our portal, where you can view based on your skill sets, and you will find the link to the job listings as well to the job listings on the portal itself towards the end. These are the links where it is all mentioned. So that was all that I wanted to mention. Now let's get started without any delay. And towards the end, I'll be taking you through the program page. I'll be discussing the schedule of this, the program, including each and every session and what we'll be covering. And also I'll showcase you the program page where you will find all the recordings if you happen to miss any one of them, and where you can also find the material, the associated resources and the supplementary material that you are supposed to go through along with each session. So that I'll be taking up towards the end. And now I would like to pass on this session to Dr. Mohit so that we can get started with today's session topics. Over to you, sir.
Thank you, Sonalika. Hello, everyone. Am I audible?
Yes, sir, you are audible and we can see your screen.
Okay, perfect. So thank you, everyone, for joining today's session. And thank you, Sonalika, for describing the program in detail and how we are going to conduct and utilize some of the resources that you showcased, and also making sure that everyone is logged in so that we can also help they can use the resources seamlessly. So now about the session, as we are meeting for the first session, I would like to introduce myself, and it'll be great if you can also introduce yourself in the chat, and that would help me also to make go through this session in a systematic way. I mean, we will be covering some of the basic concepts today, and I don't want to go much into that if you already know about it, so that we can progress towards the hands-on session that we also have planned for today's session. And we will discuss about what all we will be doing throughout the program, which will include mostly working on case studies and learning about the tools and then practicing them and then communicating with the mentors and developing a research project towards the end of the program. So there are a lot of opportunities that all of the participants will be having. And depending on your time schedule and depending on your expertise, we will be able to help you out with that. Before we start again, please, it'll be great if you can introduce yourself, like write your educational background, and if you have experience in cheminformatics or in structural biology, that would be also good to know. About myself, okay, let me play the presentation. So about myself, my name is Mohit Mazumdar, and I am a bioinformatician by education. And since last 12 years, I've been practicing and applying bioinformatics for research, and now both research and for collaborations and for education. So during my PhD, I was working on several drug targets and key proteins, as I was associated with a structural biology lab, and that had led to several opportunities to actually work on new structures which are not even published, and then what are the different ways to analyze those structures, what are this, how much they are talking about the function, and what are the different ways we can correlate structure with function, and then probably design several strategies around it. So as I was telling you that in my PhD, I was working with several key drug targets, and one of the objective was to understand the site-specific mechanism. So sometimes the objective of doing a drug discovery project or structural biology project or understanding the cheminformatics that would be required to give us the desired result, it is important to understand the specifics of the very much detailed version of what you can look at and what you can find in literature in different databases, and then develop a model. So that would be the purpose of this entire program. So understanding the site specific differences, mechanisms, comparisons, and I'm talking about three-dimensional structures here, and then developing strategies to overcome challenges specific to structure-based drug designing, and learning about what would be the next steps. So what would be the next steps for after the structure-based drug designing, and understanding where molecular dynamic simulations and quantum mechanics, all of that are applicable, and where are the innovations that are happening in the industry with AI and the deep learning. But to give you a perspective, this is a cheminformatics and biomedical drug discovery program, and we should be able to together visualize how all of this would be, because all of this will be a visual experience. So you'll be working on three-dimensional structures, and at the same time, you'll be working and looking at small molecules. So let's start with this slide, which shows the emerging drugs. So to understand that, I mean, how the drugs actually look like, it's a slide basically describing that the FDA's Center for Drug Evaluation and Research, which has approved 16 new medicines for now, and 15 novel drugs were approved in 2021, and 53 novel drugs were approved in 2020. So in 2022, the potential new drug targets are, you can see that there are big companies involved in this. You can see Novartis, AstraZeneca, and then different types of molecules which are like more target based. So if you can see that AstraZeneca's search for treatment of ER positive breast cancer, which is more specific to individual mutations, are also also described in this slide. So talking about malaria, new drugs on malaria, or the drug that Biogen came up with for autoimmune diseases, and then for Bristol Myers, the ubiquitinase mechanism targeting that, and ligase modulation. So these are target-driven drugs, and this is this is described with a mechanism of action, and we can perhaps maybe understand some of these case studies in the program going forward, that how drug purposing is also drug repurposing is also one of the ways we can counter, or we can find solutions to some of these diseases. And today in this session we will be using one of such case studies, which is a straightforward case that we will be looking at. So thank you for all of you for starting to share about your background. It's really helpful to learn, starting from Kritika, I see Harshil, I see Cynthia, I see Harish, Sashwati, and then Sana and Abigail and Broanna and Arpan and Michaela and all of you. So it's thank you so much for sharing about your educational background and where you are from. I see that we have a lot of students from participants from Nigeria, and we have a lot of also ongoing programs going in Nigeria and Africa. So good to see participants from South Africa and India. And I'm hoping that we will be able to help you with some of the structural biology related queries, structural biology related ideas, because in this program, the one of the objective is to develop that idea into a project. And that's why Sonalika specifically discussed about that in the beginning, and shared also the project submission form. So please feel free to write about your ideas. Maybe you don't have all the components there, but then you can obviously connect with us on Skype and have those discussions, and we will be able to answer them not right away, but whenever we will be able to see them, we will be able to communicate in that sense. So when you're communicating on Skype and when you're communicating in other places, please be patient, as we are working all the time, but we'll be looking at those questions and we'll be helping you out with answers. And then we have these sessions planned as well. Along with that, there are recorded sessions and videos and all of this. So I think we will be able to cover all of that. Okay, hello, Metter. So Andrea and Clement and Ishaku. So thank you again. And now about the context of the program. Now we are moving towards the actual session. So in the context of the program, this is the Learn program page, and we will be running this program for at least 3 months, which is also one of the subscription levels. So to those who are subscribing for 3 months or 60 days, 2 months or 1.5 months, 45 days, we will be able to cover and cover those objectives within that time frame. Now about today's session. So today's session is focused on some of the basic concepts that are really important to start from the beginning and develop an understanding and holistic understanding of how structural biology or computational structural biology is being utilized by researchers and utilized by industry. So understanding the molecular structure of biological macromolecules, because those are the target structures. Then understanding what are the technologies that are being used right now to generate such information, and that technology could be about the way we have evolved using X-ray crystallography, now cryo-EM and all of that. And then what are the ways to look at this? What are the primary, secondary, tertiary, quaternary and structural domains, structural motifs, protein subunits? So many of them are covered in basic textbooks, and I guess many of you are kind of aware about all of this, and perhaps anyone can Google some of these keywords and get a better understanding from the search results by going through Wikipedia and all. So our focus will not be to go through these topics just like a traditional way, but what we will be doing is, like, we will be working on some of these topics, do some hands-on and understand from a case study level. But again, we for those who are from not from a structural biology background or have never heard of some of these keywords, we have put together these lessons that will be helpful for those who wants to understand. For example, what is structural biology? Structural biology is the study of how biological molecules are built using a variety of imaging techniques. Scientists view molecules in 3 dimensions to see how they are assembled, how they function, how they interact. So that has helped researchers understand how the thousands of different molecules in each of our cells work together to keep us healthy. So structural studies have shown how mutations or mishaps molecule make us sick, and as a result, studies have prompted in new treatments for many diseases. So that is one of the objectives. What are molecules? So molecules can be grouped into two or three atoms held together by a chemical bond. So molecules, we will be discussing about RNA, we will be discussing about DNA, proteins, carbohydrates, sugar, and lipids, fat. So structural biologist, as a structural biologist, these are very particularly interest gives a lot of interest because of the nature of this to be protein, and then we are investigating these large molecules made up of combination of RNA and protein, and then RNA protein complexes. So these proteins are molecules that contribute virtually to every activity in the body. They form hair, they form fingernails, they carry oxygen in the blood, allow muscles to grow and move, and much more. So proteins are made of long strands of small molecules called amino acids, and we will be looking at amino acids, how they look in 3 dimension, and what are the way they move, and what are the different side chains and all of that. So there are 20 amino acids found in nature. Each protein contains a unique combination of few dozens of many thousands of amino acids. So some proteins consist of multiple amino acid strands that are wound together. So one of the important aspects of discussing about all of this is also like the shape. When we are discussing about the shape, we are discussing about the three-dimensional or the tertiary structure, and we are talking about the strand, twist, bends, and folds. So then we are discussing about how to understand the way protein folds. So we, in this context, would also be discussing about the new AI-driven, AI-guided DeepMind AlphaFold, and we'll discuss that how protein folding has been almost now much more easier than to do this before. So all of this, I mean, now anyone can do this, and students from high school, students without a background of biology, and specifically computational biology, will be able to learn and go through this training and learn how to apply this and how to use this. So we have a couple of lessons that describes the computational structural biology that we will be covering in this session, and the next session, introduction to cheminformatics, where we will be discussing about the chemical world. So the different aspects of proteins, like we will be discussing about these different case studies which are about different proteins. So different proteins have different functions, and their shapes or specific functions help cell hold their shapes and movement and all of that. There is a type of protein called transporter proteins that are embedded in the cell's outer membrane and form channels that helps in vital movement of vital substances such as sodium, calcium, that pass into or out of the cell. So, I mean, in the context of drug discovery or to develop new drugs, so drug typically work by either blocking or supporting the activity of specific proteins in the body. So we will be discussing an approach called structure-based drug designing, where scientists usually make a template for a protein, use that blueprint to create new medicines. So that's the mechanism in a layman's word. But to do this computationally, we will be able to do this in an in silico environment where the thought process will be to mimic what is happening within the body or within the system. So computer model would that allows us like to examine this in several ways, and that's why we were talking about molecular dynamic simulations, or we are talking about molecular docking simulations. So I mean, there are several examples now where people have determined this or have gone into different clinical stages and also come up with a drug structure-based drug, and now knowledge-based as well. So there is a lot of information and there is so much databases. And now what we've been able to do is connect that information, uh, fill up those, connect those dots, fill up those blanks, and that utilizes deep learning, a lot of deep learning, and a lot of structure based or knowledge based drug discovery. So all of this could be learned through the coursework that we've been discussing. This is a short session, so one of the objective would be to for you would be to understand this in the context that where are these resources and what we can do when before we meet again. So these are some of the things that you can do on your own, and together with the help of mentors, we will be able to work on some of these aspects. Now, the first thing we need to do to start going to get going is to understand that where is this data is available, and how to interpret this data. So today we will be doing that. So protein data bank archive information about 3D structures of proteins, nucleic acids, and protein data bank is a free repository where we will be going and doing some of our analysis, understanding where to find this information, how to interpret this information. So I was talking about the three-dimensional shapes, but in terms of proteins, you can see here in this image, different types of proteins, and like really all kind of weird shapes. And like really, I mean, that kind of explains the diversity in this area. So I mean, now what we want to know about is how complex assemblies and this understanding helps students and researchers understand all aspects of biomedicine. And we will know about application in biomedicine, which is quite evident, but it has application in agriculture, from protein synthesis to health and diseases. So the RCSB PDB builds upon the data by creating tools and resources for research and education, as well as education in molecular biology, structural biology, computational biology, and beyond. And one of the things that we will be discussing in this context is about a case study that would be on SARS-CoV-2, COVID-19. So in 1.5 year of the World Health Organization's declaration, within 1.5 year, when when WHO declared that COVID-19 is a pandemic, over 1,000 structures of SARS-CoV-2 biomolecules have been deposited in the protein data bank. It's a rare to obtain such a it's very rare to obtain such a snapshot of intense effort in structural biology, and that happened, and still there are so many structures coming up, especially with cryo EM now. And then there are other repositories. So here in the image you're seeing protein data bank, you're seeing other repositories which are maintained by EMB and EBI. So um, in that context, it's important to understand where to find the information, where to find these sort of information, which is already available, which can actually help you save a lot of days in your research. It will actually help you explain this to someone else in a very simplified way, so that you can go to the next level. So that's why that's the objective of the session. And Uniprot, for example, if you just search in Uniprot, is one of the world's leading high quality, comprehensive and freely accessible resource of protein sequences and functional information. So similarly, these are the databases and repositories which are maintained by EMBL EPI. So we will be looking at the repositories that are maintained by NCBI in the NCBI portal as well. But in today's session, we will be looking at some of these repositories. So about PDB, E is again the founding member of the worldwide WW Protein Data Bank, WWPDB, which collects and organizes and disseminates this data for bio biological macromolecular structures. So there's a lot of information structures that could be downloaded from here, and understand the chemistry, some of the chemistry, how it is connected with it. So EMBL has also developed and integrated with AlphaFold. So AlphaFold is a AI system developed by DeepMind that predicts a protein's 3D structure from its amino acid sequence. It regularly achieves accuracy competitive with the experimental structure. So I have been, talking about this AlphaFold, and in many of my webinars, we have gone through the way AlphaFold came up, and also we have gone through one of the video which explains AlphaFold. So just let me ask you this question, like how many of you know about AlphaFold, and are you interested to look at the video of how this came up, or would you want to skip that part? So can you tell me in the chat, please? So do you know about how much you know about AlphaFold? You're interested in the video, or let's skip that? So as I was telling you, that AlphaFold is an AI system developed by DeepMind, and it predicts protein structures, 3D structures from sequences. So DeepMind and EMBL, European Bioinformatics Institute, EBI, has partnered to create an AlphaFold database. So the AlphaFold database, which is what I'm showing you on the slide, is to make these predictions freely available to the scientific community. So, and since it has been an year that in the CASP 14 they did this, and then it has been a year. So since then they have done they have done 200 million predictions. So they have done 200 million predictions, which is available on this website. That is what I'm showing you on the screen. And we will be sharing the links over the email. I think Sonalika does told you about that. But after the session, there will be an email that will have all this information that we are covering, and then you will also get the video recording. So that's another way. So right. So as I was saying, that the latest this database contains 200 million protein structures and provide a broad coverage of UniProt, where you will be able to see protein sequences as well as annotation. So it's a great place to do what? It's a great place to come up with research proposals and problems and finding those proposals and problems. So you can individually download that in the context of human proteome and 47 other key organisms. So you can target organism wise, look at the structures, predicted structures, and then hypothesize from there. So it's very becoming much and much easier. It's just that one has to look at it and then start digging and then develop a proposal. That's what I will be expecting from some of you before we meet again. So please, go through these resources and then look at some of the interested protein structures, interested proteins, or interested organisms that you would want to go further, investigate further, and then we would be able to we would be able to do this. So let's move to the next video. I think you said that you are interested in the video. So in the next session, when we have the AlphaFold also planned, so these are some of the links, GitHub links, Google Colab notebooks that we will be going through. So today's session, we are not doing Google Colab. But we will be having a basic introduction to Google Colab, and then we will run some of the Colab notebooks and run some predictions using Python, Google Colab. And we'll be sharing these things with you. Now, this video, I can play this just a second. And let me know in the chat if it is audible.
We've discovered more about the world than any other civilization before us. But we have been stuck on this one problem. How do proteins fold up? How do proteins go from a string of amino acids to a compact shape that acts as a machine and drives life?
When you find out about proteins, it is very exciting.
You could think of them as little biological nanomachines.
They are essentially the fundamental building blocks that power everything living on this planet.
If we can reliably predict protein structures using AI, that could change the way we understand the natural world.
Protein folding is one of these holy grail type problems in biology. We've always hypothesized that AI should be helpful to make these kinds of big scientific breakthroughs more quickly.
And then I'll probably be looking at little tunings that might make a difference. You should be creating a histogram one and a background histogram.
We've been working on our system AlphaFold really hard now for over 2 years. Rather than having to do painstaking experiments, in the future biologists might be able to instead rely on AI methods to directly predict structures quickly and efficiently.
Generally speaking, biologists tend to be quite skeptical of computational work, and I think that skepticism is healthy and I respect it, but I feel very excited about what AlphaFold can achieve.
CAT is when we say, look, DeepMind is doing protein folding, this is how good we are, and maybe it's better than everybody else, maybe it isn't.
We decided to enter CASP competition because it represented the Olympics of protein folding.
CASP, we started to try and speed up the solution to the protein folding problem. When we started CASP in 1994, I certainly was naive about how hard this was going to be.
It was very cumbersome to do that because it took a long time.
Let's see what are we doing still to improve.
Typically 100 different groups from around the world participate in CASP, and we take a set of 100 proteins and we ask the groups to send us what they think the structures look like.
We can reach 57.9 GDT on CASP 12 ground truth. CASP has the metric on which you will be scored, which is this GDT metric.
On a scale of 0 to 100, you would expect a GDT over 90 to be a solution to the problem.
If we do achieve this, this has incredible medical relevance.
The implications are immense, from how diseases progress, how you can discover new drugs. It's endless.
I wanted to make a really simple system, and the results have been surprisingly good.
The team got some results with a new technique. Not only is it more accurate, but it's much faster than the old system.
I think we'll substantially exceed what we're doing right now.
But then it's a game changer, I think.
In CASP 13, something very significant had happened. For the first time, we saw the effective application of artificial intelligence.
We've advanced the state of the art in the field, so that's fantastic, but we still got a long way to go before we've solved it.
The shapes were now approximately correct for many of the proteins, but the details, exactly where each atom sits, which is really what one would call a solution, were not yet there.
It doesn't help if you have the tallest ladder when you're going to the moon.
We hit a little bit of a brick wall since we won CASP. Then it was back to the drawing board and like, what are our new ideas? And then it's taken a little while, I would say, for them to get back to where they were, but with the new ideas. And then now I think we're seeing the benefits of the new ideas. They can go further, right? So that's a really important moment. I've seen that moment so many times now, but I know what that means now. And I know this is the time now to press. We need to double down and go as fast as possible from here. I think we've got no time to lose.
So the intention is to enter CASP again. CASP is deeply stressful.
there's something weird going on with the learning, because it is learning something that's correlated with GDT, but it's not calibrated.
I feel slightly uncomfortable. We should be learning this, in the blink of an eye. The technology advancing outside DeepMind is also doing incredible work.
And there's always the possibility another team has come somewhere out of left field that we don't even know about.
Someone asked me, well, should we panic now? Of course, we should have been panicking before.
It does seem to do better, but still doesn't do quite as well as the best model. So it looks like there's room for improvement.
There's always a risk that you've missed something, and that's why blind assessments like CASP are so important to validate whether our results are real. Obviously, I'm excited to see how CASP 14 goes.
My expectation is we get our heads down, we focus on the full goal, which is to solve the whole problem.
The Prime Minister has announced the most drastic limits to our lives that the UK has ever seen in living memory.
I must give the British people a very simple instruction. You must stay at home.
We were prepared for CASP to start on April 15th because that's when it was originally scheduled to start, and it's been delayed by a month due to coronavirus. I really miss everyone.
I struggled a little bit just kind of getting into a routine, especially with my wife. She came down with the virus. I mean, luckily it didn't turn out too serious.
CASP started on Monday.
Can I just check this diagram you've got here, John? This one where we ask ground truth. Is this one we've done badly on?
We're actually quite good on this region. If you imagine that we hadn't have said it came around this way, but had put it
down instead. Yeah.
One of the hardest proteins we've gotten in CASP thus far is a SARS-CoV-2 protein called ORF8.
ORF8 is a coronavirus protein. We tried really hard to improve our prediction, like really, really hard. Probably the most time that we have ever spent on a single target.
So we're about 2/3 of the way through CASP, and we've gotten 3 answers back.
We now have a ground truth for ORF8, which is one of the coronavirus proteins. And it turns out we did really well in predicting that.
Amazing job, everyone, the whole team. It's been an incredible effort.
Here, what we saw in CASP 14 was a group delivering atomic accuracy off the bat, essentially solving what in our world is 2 problems. How do you look to find the right solution, and then how do you recognize you've got the right solution when you're there?
All right, are we mostly here? I'm going to read an email. I got this from John Malt. I'll just read it. It says, John, as I expect you know, your group has performed amazingly well in CASP 14, both relative to other groups and in absolute model accuracy. Congratulations on this work. It is really outstanding.
AlphaFold represents a huge leap forward that I hope will really accelerate drug discovery and help us to better understand disease. It's pretty mind-blowing.
These results were, for me, having worked on this problem so long, after many, many stops and starts, and will this ever get there? Suddenly, this is a solution. We'd solved the problem. This gives you such excitement about the way science works, about how you can never see exactly or even approximately what's going to happen next. There are always these surprises, and that really, as a scientist, is what keeps you going. What's going to be the next surprise?
All right, and that was about the AlphaFold discovery and their team and how they able to work out and come up with this algorithm. Now, in context of how we are going to use it, it is a different story altogether. So what we would be trying is, there are some, obviously this is a huge leap and has helped us in finding some of the those structures where we have the information before, but where there is no information, where there is no experimental structures available, and if there's a unique motif or protein, that is something that is still not answered by the AI-based models. So some of the limitations that would be answered through experimental techniques, and we have those techniques which are evolving also at the same pace, and with the new structures and everything, I think this is going to be getting better and better only. The last database today that we're discussing about is the ChemEMBL. So ChemiMBL is another, now we are discussing from discovery now to chemistry. So ChemiMBL is a manually curated database of bioactive molecules that has drug-like properties. It brings together chemistry, bioactivity, genomic data as well to aid the translation of genetic information into these effective new drugs. So ChemMBL is a database again which has two-dimensional structures, calculated properties such as log P, molecular weight, and then Lipinski parameters, and abstracted bioactivities such as binding constant, pharmacology, ADMET data. So this data is abstracted and curated from either publications like primary scientific literature, and covers fractions of these and helps us in structure, activity, relationships, and discovery of modern drugs. So as we have been discussing and discussed so many these, not so many, at least 4 or 5 of these databases, it'll be really great if you can maybe browse some of them, as we will be also sharing with you the links and all after the session. So please, after the between the sessions, please go through this and have the questions that you can ask us related to them. Now we will be moving towards the case study, and we discussed about coronavirus and the SARS-CoV-2. So we'll take SARS-CoV-2 as a case study. So the entire all of the SARS-CoV-2 pandemic has also shown a light on many diverse contribution of this in silico structural biology work. So initially, structure prediction work we used to use, we had used in that time modelers, iTesser, and AlphaFold, and this DeepMind, all of that. But one of the key challenges now is to understand that you heard of the way some of these proteins replicate, and the way they create these mutations, and there are several copies. So understanding the way the mutations, actually these mutations can disrupt or become more lethal. So we have a case study planned on that, not in the first session, obviously, towards the second or third session, where will be able to look at the specific types of these variants and the structures, because many of the structures have now reported and at a very good resolution. So we can do that case study where we can compare the variations that you could see in different types of these variants where there are different mutations at different locations. So that is the case study that we have planned. But in addition, like if you look at the structures, all of this started with the spike protein, and which is at the surface of the coronavirus. So these spikes, and giving them their distinctive crown-like appearance in electron micrographs, that's the reason they are called spike. So the spike initiates the process of infection, and then binding to the receptors, and then fusing with the cell membrane to release the viral genome inside. And many of you might be are virologists and have a better understanding. But from a structural point of view, what I'm trying to describe here is that many other enveloped viruses, such as similar to spike proteins, use the similar mechanism to infect cells. That has been known. For example, influenza and the enveloped glycoproteins of HIV-1 and Ebola as well. So this the spike protein is composed of 3 identical chains. So we are talking about 3 chains, A, B, and C chains, and then together they form a complex with a small domain inside the virus, a membrane spanning segment, and a large ectodomain that extends the spike protein into different types of, like it is very flexible to move in that manner. So in this slide, I'm trying to explain the same thing, that when you look at these structures, and recent structures have revealed that the spike of SARS-CoV-2 and SARS-CoV are kind of flexible. So in this structures, the receptor binding domains are seen in different conformation. Now, about conformation is again one of the limitation. With the same protein, you will find different conformations. So how do you understand that what would be the best conformation to target, or what is the actual or the average conformation and all of that? So those are some of the questions that could be answered when you go through and learn from different aspects about molecular dynamic simulations, averaging structures, and understanding the movement. But actually, these different conformations actually helped us a lot with all of this. The extended conformation is needed to bind the receptor, and and um, and the flexibility is a great advantage actually to the virus, because the researchers hypothesized that the recent SARS variants are virulent because the receptor binding domains are particularly flexible, whereas the more widespread coronaviruses that causes the common cold, which are less flexible. So these are some of the examples that we should be able to take a look at, and let's do that in the context of visualization. So this last part of the session is doing some hands-on using molecular visualization. So that's, as we have shared that with you. So 6M17 and 6VSB are the 4-digit letter code that you can find on PDB. So let's click on some of these links and open this in a browser, and let me know if you can see my browser or not. So this is a link that where we have opened a PDB file, and then in the context of visualization, we are going to use PyMol. So PyMol is a strong molecular visualization software, and in this slide you will be able to see that I have put together a lot of links from like how you can install PyMol. So can you tell me, like, is it installed in your system, any of you? Like, do you know, like, have you installed PyMol ever? Or is it like something that you can do? So, all right. So many of you are saying yes, you have it installed. And for those who haven't or hasn't, I'll share these links in the chat. Perhaps that would be a quicker way. We will be sharing this in an email, but let me share all these links in the chat. So those who have not installed can utilize. And one of the thing is that PyMol is a proprietary software by Schrodinger. But again, there are academic versions, educational versions available. They are a little tricky to install, but you'll find one of them in the link. So if you don't have the full version, or if you have the demo version, and if you use it for academic purposes, you can have the full version and all the capabilities, exploring some of these links. And I think that we will not do in the session, but if you have issues, please feel free to write that in the chat group, and we will be able to help you. Now, if you have installed it, the next step is to use it. And before using it, let's understand a little bit about the interface of PyMol. So the interface, before understanding about the interface, it would be good to understand that what are the basics, like how do we visualize files and what is there in the PDB file. So can you tell me, like, do you know what is there in a PDB file, how the coordinates are stored, or do you have any idea like about the X-ray crystallography and all those methods that generates these experimental files? Because what I want to discuss next is this text file that we get from the experiments like X-ray diffraction, X-ray crystallography, or other techniques. So this is the file, this is a text file. So when you're downloading from PDB, you kind of download a text file, and then you open it up in a software that kind of projects these coordinates in a three-dimensional space. Does that make sense to all of you? All right. So that's the whole idea, that from a text file, the software generates the coordinates and put it in a three-dimensional space. And I'm discussing this because it's important to understand, and if you understand this, you will be able to actually do some modifications on your own just through the structural visualization. So what we want to understand in this image that is on the left hand side of the screen is the PDB file. Yeah, we can do it, Girija. So let's do this practically. I'll stop we'll go to that website and let's do this there. So in the context of this file, and this is a structure that I've been describing in the presentation. It's a membrane protein and it's a viral protein. And there's this information that this is generated through cryo-EM technology, and this is the resolution. Now, there are other information. There's an associated publication published in Science. So you can check the article here and understand more about it. That would be the best way to understand about the structure. And then there are different information. So this is the complete A chain and D and C chain. So we have the sequence is 654 amino acid long, and this is one of the visuals of the structure. And then going down, you will find more information about different clusters, and then you can zoom in and look at these individual residues, compare these different sequence clusters, which are similar to this protein. And then the second molecule. So this is like not just one molecule, there are two molecules. So angiotensin covering enzyme 2. So ACE2 receptor is also there. So you will be able to see the protein-protein complex in this molecule. Now, similarly, there is a third molecule, the spike protein S1. So this information is right here, like the number of molecules that are there in the three-dimensional structure, co-crystallized. There are so many co-crystallized structures. Some of them are part of the experimental structure. Some of them are artificial artifacts from the solution. So you have to understand that which are the important part and which are not. And this sort of information is available right here. That's the way to read through this information here, that once you go to this page, read about this information, get that understanding. And there are here tabs also. So 3D view, and then annotation. So we can go through all of this. This will take a while, but let's download this structure first, and then we will do a visualization on PyMol. And the next time then when we meet, we will be able to maybe discuss all of this and answer the questions that you have, and you'll be able to ask those questions. So you will see that there are different ways that you can directly visualize right here on the browser. So you don't even need to have PyMol for that. PyMol is for those maybe where you're predicting and you're modifying. So it's great to have PyMol capabilities, generate high resolution structures. But here also these capabilities are there. You can look at active sites, you can look at different sites, you can click and understand the interaction also, which is a great interface that PDB is now providing within this portal. So I hope that you all are going to utilize some of these links, and I'll put it in the chat, especially this one that we are looking at, so that you all can look at it, and we can maybe do it together. So now let's download this structure. So while downloading these files, you'll find different file formats that are there. Now, PDB format, there is this MMCIF format. So we can discuss about these formats, but let's look at some of the one of these formats. So I've downloaded this file, and I can open it in a in a text editor. So let me open this file in a text editor. So you can use Notepad, Notepad++, WordPad, any of these text editors to open a PDB file that you have downloaded now. So using the link, as you saw that I easily downloaded that file. And this is how it looks. Like this is the structure file. This is the maybe a receptor file. This is how it looks. It has a lot of remarks from the top towards going towards the end. And these remarks are not read by the software, but they explain the characteristics of this molecule or the structural factors and some of the chains, some of the interactions, as well as these sort of information. So there's a lot of information this gives. Just to tell you in a nutshell, so header, title, author records provide information about the researchers who defined the structure. Numerous types of records are available to provide other type of information. Remark record provides contains free form annotation. And then sequence res records give the sequence of these three peptide chains like A, B, C, D, and all. Atom records. Now, what is important here is the atom records. From here, this is the important section. This is the actual file. This is the actual structural file. So this is atom. This atom describes the coordinate of the atom that are the part of the protein. For example, the first atom line describes the alpha N atom. So this is the nitrogen at the alpha position of the first residue of the peptide chain A. I hope that this is kind of clear when we will look at it in a three-dimensional visualization as well. But that is how it is. So it is a valine, and the residue is valine, and the first three floating points, this is X, Y, Z. This is X coordinate, this is Y coordinate, this is Z coordinate. So X, Y, Z will be transposed or will be plotted in a three-dimensional software called PyMol. Now, I think this is clear, and this is kind of, I explained it in much more depth. The next three columns are occupancy, temperature factor, and the element name respectively. Then you will find towards the end, there are different chains. Like I'm scrolling, and I hope all of you are able to see this. So this is A chain, as we know that this is a big protein. Then there is B chain. You see that A has changed to B now. Now this is B chain. So another subunit. And then there will be other chains also. E chain and C chain. As we were reading the molecule there, D chain. And then after the atoms, I just wanted to show heteroatoms. These are the heteroatoms. Now, the ligands are usually heteroatoms. So here you will find these NAG, which is like a crystallized, I think these NAGs are present. These are, sorry, not crystallized, like there are some conditions for this in the solution. And then they are there. And then there is water molecule, and there is zinc also. So here towards the end, you will find zinc, water molecules, and all of that. So each chain had some water molecules that have been traced and put together here. So this is how you read the PDB file. Now you can manually actually extract some of this information and paste it in a new file and create .pdb. So you can simply, like if you want to extract the like the F chain, you just cut it like properly and paste it in a same format and save it, like, and rename it like fchain.pdb. So that way you can manipulate this PDB file. You can take some of the segments and open it and do those exercises. And those will be the assignments for today's session, to manipulate my PDB file and to do this on your own. Now, the next thing we are going to try is PyMol. Now I have installed PyMol on my system. Now I'll open PyMol by this command, PyMol. and this ampersand sign is actually a sign that let me use the console. Now that's not important. So yeah, PyMol, that is what we would be interested in. So we have opened PyMol and we have downloaded a structure. Now let's open that structure in PyMol and let's look at it and understand what are the different ways we can visualize it. So now I have opened it in PyMol and I'm rotating it using my key mouse pad, tracking pad. Now let's yeah, I think it's visible to all of you. Now here there are a few commands, A, S, H, L, C. So A stands for action, and there are two tabs here, all and this one. So you can actually load multiple structures, and in the next session we will be discussing about structural alignment, structural comparison, and there is a workshop also coming up on AlphaFold and doing all of this. So I hope that as many of you would join the program, will be coming and participating on all of those events. Now looking at the structure, now you can zoom in as much as you want, and you can dive and look into each molecule. So here I've just selected, clicked here, and there's one atom which is selected. Now you can see that there are dots, but it is not showing in the presentation because the presentation that we are looking at is the cartoon presentation. Now there are different types of presentations, visual presentations that we will be looking at, that we'll be exploring, but this is a cartoon presentation. Now to look at the side chain, you need a stick presentation. This is a stick presentation. So this is the NAG molecule which is plotted all over here, which is you can see all over the place, NAG molecule. But I have clicked on an amino acid which has a ring, as it seems. Now I want to see what is this amino acid. So what I will do, I'll go to the select, because I've selected it. There's a new tab that has come up which says SELE, which is select. This is selected. Now whatever I'll do will make change to only this area. So this is the select, and I'll do what? I'll try to show stick. As I was telling you, this could be seen in a stick representation. Now you can see it's a stick representation, and you can look at the residue. You can take a look at the residue. Now, this is important in the context that if you want to know about that, if there is a mutation and you have figured out that you know that there is this mutation, so you want to see what is the effect of the mutation, and you can check out that by looking at the residue, and then selecting that residue like it is selected, and in action I'll use this function called modify around 4 angstrom. So I wanted to see that where this residue is interacting with, is it interacting with any other other intramolecular interactions within the protein. So let's look at that, because if we want to change this, if we want to mutate it, obviously some of these interactions will be lost. So as I did that, what I did is that I went in action, modify around 4 angstrom. So I checked what are the interaction around 4 angstrom. Now I have other residues also getting highlighted, or there are these selections. Selection has expanded to other molecules, which means that there are other molecules that are there interacting. Now I can also manipulate the structure and mutate it. This is what, let's take a look at what is this side chain. So to name it, there is this tab that says label. So this is label. Here you can also written action is written, show is written, hide is written. So you can show something, you can hide the same thing. This is label, it's written, and then there is color. You can change the color to different colors of your liking, or to highlight it basically. Now I have selected this. Now I want to mutate it, but before that I need to know which atom is it. But this is tyrosine. You know this is tyrosine because you can see here when I've selected, this console is also getting updated, and it's telling me that this is a B chain tyrosine at 641 position. But we can label it, and she see it in here. So we label it. It's showing that now in the screen. You can see it's so showing TYR 641. Now I want to change this tyrosine to alanine. How would I do that? How can I do that in Pymol itself? So there is this plugin or wizard. There is this mutagenesis that you can do here, right in here. Right, it comes up here. Now I have selected this one, and I want to change this mutation. I want to bring a mutation in the structure by changing tyrosine to alanine. Now, what do I want to change it into? Alanine. So I'll put ALA, and I'll apply. So I've changed mutate to alanine, and these are some of the other options that I can change. So I'll apply. It's asking me to pick a residue. So let's pick a residue. All right. So let's try this again. It's asking me to select the rotamer. Okay, so I think there is a library file for rotamers that we have to put in here, but there is this mutation. I think this now has changed into alanine. So the side chain has gone, but I think with the rotamer library, it will be like give us probabilities of this mutation. So now this mutation it's changed into alanine. So this is one of the way I was just showing all of you to work on these protein structures and do some manipulation using Pymol. Now, what we did is that we downloaded a structure, we played around with it, and we learned about some of these things. Now, for the assignment, for the students or participants of the program, we'll be sharing some of these protein IDs and ask you to prepare a report where you have done some of this work, or we can discuss any of your interest for what we have covered in this session. Now, as we learned about Pymol, we learned about PDB, we learned about different databases, and we also learned about how can we do some of these manipulations. Now, we can go much depth of these, and we will be using these tools throughout the program. This is just an overview so that many of you get started with these things and develop these expertise throughout the program with the help of the mentors. Now, going back to the presentation. So in Pymol, what we have done is we have done the installation, and then we have done some basic analysis. Then another visualization software that is there is Chimera. And in the next session, we will look at Chimera, and we will look at some of the capabilities of Chimera, and which is utilized for. Similarly, we will do mutagenesis, and we will do some RMSD calculations where we will compare two structures and see how much they are different. And then we have these sessions coming up, introduction to Cheminformatics in the next session. Then we have chemistry and protein databases, many of which we have covered, but we'll go in more depth. These are some recorded sessions also. And then we have the protein structure prediction, some of the case studies like docking experiments setup. What are the different, as we were discussing, the more we are being able to mimic it, the more high probability of this to be accurate. And then the drug repurposing case study that we were discussing will be discussed as well. And then the advancements of transformation using GPU computing and deep learning, and different molecular generation using deep learning. So these are some of the things that we have it put together. And before I pass it on to Sonalika to discuss about the other aspects, I would request you to join LinkedIn, this group. This is a group where this has grown now more than 20,000, 2,000 people. So here we frequently post about the job opportunities and all of that. And in the OmixLogic group on Facebook, we also do the same. So if you're on Facebook, LinkedIn, please join these groups, and then you'll be able to find job opportunities there as well as on the Learn OmixLogic portal. So that's what I wanted to cover in today's session, and I'll be happy to take the questions that are there in the chat if Sonalika or you can let me know about those questions. And in the meantime, it was great meeting you all for the first session. I hope that this was something that was helpful and you learned something new, and we will try to make this more and more interesting throughout the program. Thank you so much.
So we have a question by McKenna. She's asking, what is the purpose of seeing the activation energy while talking?
What is the purpose of seeing what? Sorry, can you repeat
what is
that?
the purpose of seeing the what is the purpose of seeing the activation energy while talking?
So activation energy, I mean, are we talking about energy minima and maxima? Like sometimes molecules are in different energy states. But what we do usually in our calculations is that what we do is we kind of do energy minimization. So energy minimization, what it does is that it puts the structure in a global low energy state. And that is said to be more close to the native protein. And that process of doing energy minimization, the steepest descent and conjugate gradient, these are the algorithms. And there are different softwares where we can achieve this molecular minimization, energy minimization. And that helps us to get to low potential energies and negative delta G. So the changes you can see is that when you look at delta G, you'll be able to look at you'll be see that there's very high delta G. And then when you run energy minimization, you'll be able to see that the delta G has changed and it's more now stable. And what happens actually is that the atoms their location and all of that, they are just like changed based on to the most acceptable conformation. That's how the software, the algorithm works. But yeah, but that's I hope that answers your question. Sorry, Sonalika.
So then we have a question by Mithi. Hello. So can you hear me now?
Yeah.
Yes, sir. So, sir, a question by Miti Mathur. She's asking, is AlphaFold server better than already available protein structure prediction servers like iTaser and Modeler?
I think it's good. It's a great question, but it actually changes from study to study. So some proteins, I mean, I have done AlphaFold, and I will not say that it predicts everything like Dream or like it says it does, because there are other challenges also. But yeah, I mean, iTester and models are equally good. So it's good to have like a maybe comparative study while you go for a structure prediction. Do like all of them and see and try to look at the statistics and decide.
Thank you, sir. I hope that answers your question, Miti. And we have some questions regarding assignments and also the session recording, I would be able to answer those questions just in a while while taking you through this program page. So if you have any technical questions for Dr. Mohit in the meantime, you can definitely put in the chat box. And here is the question by Harshal. He's asking what would be a better choice of force field and energy minimization combination?
Yeah, I mean, I like the force field that Schrodinger has developed. It is nice. The amber force field, the charm force field. I think all of them are good. Like all of the ones that have been published, and like thousands and thousands of citations have already happened for those. So you want to take for predictions and dynamics and all of that. I mean, it's advisable to use something which is validated over and over. And I mean, that's my recommendation.
Right, sir. Thank you. All right. So now going back to the program page. I've shared the link of the Cheminformatics program page in the chat box. This page is very important for all those who have joined the program, for you to go through the schedule, keep a track on what schedules, what sessions do we have scheduled on which particular dates, what are the associated resources, and we will also post the recordings of the mentor guided sessions here on the program page itself. So it's very important for you to keep a track on this program page for the entire duration of your enrollment. All right, so today we had the first session. Those who are asking about the recording of the session, the recording of the session will be updated on the program page in an hour or two. Just when the processing of the recording is complete, we'll upload it. You can then access the recording. The program page can be accessed by the program participants. So if you will have a badge on your profile named as Cheminformatics for Biomedical Drug Discovery, if you click on that badge on your profile, you will be redirected to this program page. I'll just show it to you shortly. Or you can directly access the page with the link that I've shared in the chat. I'll also share this link via an email. So you'll have this link with you where you can access the recordings, look what the resources are that you have to follow, and find the links of the supplementary material as well. So here we have the session schedule that I was talking about. Today we had the first session. The next session is scheduled for August 25th. So we'll be having sessions for this program on every Monday and every Thursday. All right. Today, the first session was an exceptional case where it is scheduled for Tuesday because we already had some pre-scheduled sessions scheduled for yesterday. But otherwise, you'll have these sessions on Mondays and Thursdays to keep a track of these sessions. That's what I'm telling you. Then we have the third session on chemical and protein databases on August 29th. We have mentioned the topics here. I won't be going into too much depth of each and every topic for the sessions that we would cover. Otherwise, it will take a long time. Then we have the fourth session on target structure prediction on September 1st. Target identification virtual screening we'll cover on September 5th. Practical experience in docking, we'll have a hands-on session for this on September 8th. And on September 12th, we'll have the session on data science and AI in healthcare. And we'll practice the R Collab notebook for the same. On September 13th, we have a workshop on AlphaFold, a 2-hour-long workshop where we are going to have a practical hands-on AlphaFold. All right. And this is what it is mentioned in the program as well. This is a 2-hour workshop specifically. On September 15th, we have the next session on recent advances in drug discovery. This session will be taken up by an industry academy expert. And then you have the next session on AI in molecular drug discovery on September 19th. The next session is scheduled for September 22nd on advanced cheminformatics. And then towards the end, we'll be discussing about how you can design a cheminformatics research project and get started altogether. And finally, discuss some case study examples of successful and failed drug development by industry experts who were involved in these successful and failed drug development processes. All right, so that is what is the schedule of this program. You can find it on the program page. And today was the first session, and we invited also the participants who were interested to enroll for the program, as they wanted to get some insights how the session would be conducted. Today was a much introductory session because we wanted to get you all started and introduce you to all the resources so that you don't skip any one of these. Please make sure that you join the Skype group because that is the way we can stay connected every time. Yes, mail is also a very good option where you can mail us on support@pine.bio and connect with us for any query. But joining Skype will also keep on updating you about the sessions, recordings, resources, supplementary material that we share, and also have a chat with the mentor regarding the questions that you might have or ask for a live Zoom meeting to clarify your questions. All right, so it would be very helpful. Please try to download Skype. If you're not there on Skype, create an account. We'll share the link with you. Please join the community. All right. Now, for those who have not yet joined the program and were here just to get some insights on how the program will be conducted and what the structure of the program is, but wish to continue for the other sessions, they can register themselves for the program. We are accepting registrations till tomorrow. And here we have 3 different subscription types that you can pick and enroll for. It's completely on you which particular level type you want to pick. I'll just quickly explain what these includes. So these different subscription levels are specifically or will give you access to the program and the resources for different numbers of days. That is what it means. The 45 days one will give you the access to the whole training program. A point to be mentioned, the resources in all of these 3 different subscription level are the same. You'll get the same session recordings and the supplementary material as well. There is difference only in terms of mentor guidance and research work that you do or that you work on. So in the 45 days, you will be able to just complete the training program. But in 60 days, as you will have some more time, you can meet the mentor, clarify your questions, and also discuss your research question that you would like to work on, design your research hypothesis with the mentor in these live sessions. So it will include more of interactions with the mentor where you can get all of this done. And then the 90 days access, that is the advanced level subscription type. You will be able to complete a research project along with the training part as well. This is what I explained in the very beginning of the session, where I was saying that the participants who have taken up the 90 days of subscription, you are required to work on a research project. So the moment the training process is complete, the moment the training part is complete, all the sessions are covered, all the practical hands-on sessions are completed, and till then you'll have a good idea about the concepts, and you will have some good hands-on practice as well with these tools and softwares that we'll be covering. All right, along with these sessions, what you can do is you can start a literature review and at least come up with the research question that, or maybe have some idea at least that you would like to discuss with your mentor. So you can then schedule a meeting with your mentor, discuss your idea, what exactly you want to work on, and the mentor could help you frame your question in the right manner or modify a question if it is required, and then you can get started to work on your research project. All right. Now, what would be the outcome of this research project? The very first thing is that you will get to publish this project on the OmixLogic Learn portal itself, where you will have this research publication that you can share with your community or future recruiters, or if you are a student, a graduate or a postgraduate student and want to pursue higher studies, you could use this publication for that as well. You can present this research work in any good conference or symposiums in the poster presentation format. All right, and definitely proceed ahead to publish your research work in a reputed journal as well. So that could be the 3 different outcomes of working on a research project. That was it that I wanted to discuss. Now it's up to you which particular subscription type you want to pick. It really depends on how many days do you have your availability for to work or to learn a new skill set and work on a project. All right. And also we are offering great scholarships to students and researchers. So you can reach out to Sparsh on marketing@omixlogic.com, and she'll be able to help you out with the scholarship opportunities. She'll be able to tell you how much scholarship can you get on the program. And also, Sparsh, if you can share the scholarship form so the participants can fill that out. We'll review your application and we'll get back to you on the scholarship that you can get for joining the program. So that was it. Now we have some messages in the chat box. I would like to address them. Did I've explained 60 days? So I think that is clear to you. Yes, definitely. You will get the certificate. So where do you get the certificates for the courses that you complete and for the program that you complete? So when you will complete all the sessions on the program page that I just showed you, after the completion of all these sessions on the program page, you will get a certificate automatically on your profile under the programs tab. Let me look for a program that I've completed so that I can show it to you. Here it is. There was a program on Introduction to Cheminformatics in the month of April. So I completed the sessions, and then you can move your certificate from here, and you can also download the certificate directly from your profile section itself. That was about certificates, and also along with each and every session, as I told you, that you have to complete some associated resources, which are the courses on the OmixLogic Learn portal itself. So also for each and every coursework that you complete, you are going to get the certificate that you can download and share your achievement on social media platforms. And Sparsh has now shared the scholarship form in the chat box. Thank you, Sparsh. Those who are interested to avail the scholarship, please fill out the form, and our team will get back to you after reviewing your application. Zee Mehta, you were asking about how bioinformatics can help you in your further studies. So for that, it is first of all very important. As you are a biotech student, it's very important for you to first of all know what the field of bioinformatics is all about and identify a research interest. All right, so for that, you can go through the bioinformatics coursework and Bytes and Molecules on the OmixLogic Learn portal. These two courses are free courses. You just need to create an account and directly start with Introduction to Bioinformatics and Bytes and Molecules. And these courses will give you a good idea about what bioinformatics is all about, what is the role of bioinformatics in healthcare, and what are the different types of big data or omics data that the researchers are now dealing with or working on. All right. So it's a very short coursework which will help you get a good understanding. And then you have the next coursework, that is Bytes and Molecules, which will help you understand the different types of omics data types, I'll say, or fields like genomics, metagenomics, transcriptomics. Because you are a BSc biotech student, I know you are at a very early stage. So you can go through Bytes and Molecules and get a good idea about what are these different types of omics data types and what are the different specialization tracks where these different omics data types have their applications, and what type of data analysis is involved and stuff. So these are introductory courseworks which are definitely free of cost. You can just simply sign in and go through them and identify your interest first. Like identify your interest, which field interests you the most, and then you can take it further and learn more about. All right. So today we are talking about structural biology or cheminformatics. So there is a basic coursework that has been recommended for you all to get started for today's session, that is protein structure and function. So you can go through this particular lesson where we have tried to explain what are the different types of protein structures, and also how the structural biology was evolved all together, and what are the basic concepts involved. All right, and Cynthia, you will get to know about the bioinformatics tools for computer-aided drug designing, definitely, or cheminformatics, various tools and databases throughout this program. Today, our idea was just to introduce you to these different tools and databases. Now we will take each one of them and dig more, get in the upcoming sessions, we are going to get more insights about each and every tool and software or database. We'll explore these databases, extract these structures from these databases, and then work on them using these different types of tools and softwares. So today was just an introductory session. We wanted to keep it light and not overwhelm you all a lot with all these different types of tools and databases and softwares working. How do they actually work? But now we try to move from a beginner level to an advanced level slowly. All right, I think those were the questions that we had in the chat box to address. And if there are any other questions, you can please put in the chat. Oh, Cynthia, about the 60 days part. So I said that you have 3 levels, 45, 60, and 90 days. 60 days means 2 months, all right? So you will be completing the training part in 1.5 months, and the other half month, or let's say 15 days that you will have with you, you can discuss with the mentor your research hypothesis, all right? You will get to design a research project, okay? And to get started with it or to work on it, you definitely need an extension. So a 90 days of project will definitely assure a publication, a research publication. But on a 60 days, if you take up a 60-day subscription level, it won't be easy for you, because in 2 months, you can't complete the training also and then work on a project and get a publication. That's practically not feasible at all. So you will complete your training part first, and then do the literature review, come up with a question, design your hypothesis, and then you start working on a project. So if you want to come up with a publication, a research publication out of this program, then maybe you can take up the 90 days or the subscription level. And if you're seeking scholarship, then Sparsh can help you with that. So Bridget, the first 2 courses, Introduction to Bioinformatics and Bytes and Molecules, are free courses because these are introductory courses, and there are many people who come from different fields. They might not be from bioinformatics, they might be from biotech or microbiology or biochemistry, or a completely different field like biophysics altogether. So these 2 courses are for them to get to know or get introduced to bioinformatics and different omics data types, introduced to data science, and get introduced to structural biology. That is why these 2 courses are free. And then to access the other courses, you have a license involved, because in other courses, you have the data analysis practical sessions involved as well, which require you to take up the access of the T-Bioinfo server, where you carry out all the practical hands-on analysis. And on the server, you will be allocated 5 terabytes of space. So for the space and the license, it actually requires a specific fee to maintain these particular spaces and licenses that we are providing. All right. So introductory courses are free, of course. You can go through them and then identify a research interest and then take it further. I hope I was able to answer your question, Bridget. And do we have any further questions? All right, thank you. So if we have no more questions, we can end today's meeting. Thank you, Clement. We can end today's meeting, and we'll be now meeting on August 25th for the second session. I will be mailing out the recording of the session, the resources that you have to go through, and the assignment that you have to complete. All right. And also, I will share the link to join the Skype group. Please make sure that you join the Skype group. If in case you face any technical issue in joining, please email us at marketing@omixlogic.com. And for the participants of the program, please make sure that you have an updated profile. We require you to have an updated profile so that we can monitor your progress. All right, Briona, please make sure that you have an updated profile, and also the other participants. Okay, that was it. And you need to submit your assignment on marketing@omixlogic only if you're not a program participant. If you are a program participant, then please email us on support@pine.bio. Okay, that was it. And hoping there are no more questions, as I can't see in the chat box. We are good to end today's session. Looking forward to